Sometimes siblings look incredibly similar. Other times, they can look very different. This is due to the process of meiosis. After learning about mitosis and cellular division, it's also important to talk about meiosis, which is the process by which our gametes are formed, or our sex cells, sperm and eggs in mammals. So gametes have half the number of original chromosomes as the original cell. And again, this is important. So when the sperm joins the egg, that fertilized cell has the correct amount of chromosomes. So in humans, a haploid cell, a sperm or an egg, has 23 chromosomes, and a diploid cell, so a somatic cell or a fertilized cell, has 46 chromosomes, 2 copies of each individual chromosome. So the phases of meiosis are named the same as the phases in mitosis: prophase, metaphase, anaphase, and telophase. But since in meiosis it goes through these phases twice, they have numbers. So prophase 1, prophase 2, metaphase 1, metaphase 2. And different from mitosis, in metaphase we produce 4 daughter cells, and none of them are identical to each other or to the parent cell. So after meiosis, we can have the sperm and the egg formed, and then the union of those will form a zygote, or a fertilized egg. And a zygote contains one of each of the biological mother's 23 chromosomes and one of the biological father's 23 chromosomes. So this is why people have a mixture of characteristics from both of their biological parents. So these paired chromosomes are called homologous chromosomes because they're similar in size and shape, if we remember that karyotype from a couple lessons back. And if development proceeds normally, the zygote will begin developing by mitosis. So it'll go from single-cell zygote to 2 cells to 4, 8, 16 morula, following on to a blastocyst. So meiosis occurs in the ovaries to form ova and in the testes to form sperm. And there's 2 rounds of cell division, meiosis 1 and meiosis 2, to produce 4 haploid cells. Now, recalling back to human reproduction, remember that 4 sperm cells are produced. And in ova development, 1 viable egg is formed, and then 3 non-viable polar bodies are formed. And they all have the same amount of genetic material, but they have different function in egg division. So before those meiotic divisions occur, the cell goes through interphase just like in mitosis, and in order to replicate the DNA and have the cell grow. So here is cells in interphase. Again, we have the nuclear envelope and a lot of chromatin visible in there. And in the S phase of interphase synthesis, the genetic replication occurs. G1 phase is growth, and then G2 phase is also growth, but other enzymes are readying for cell division. So meiosis 1, that cell division is actually pretty similar to mitosis. It involves the 4 following phases: prophase 1, metaphase 1, anaphase 1, and telophase 1. So in prophase 1, that nuclear membrane begins to dissolve, the centrioles split and move to opposite poles, and spindle fibers are established. So here we can see in the diagram, we have our centrioles moving to the poles, but unlike the diagram that we had in mitosis, they're not exactly at the poles yet. But similarly, you will see the nuclear envelope dissolving, as well as those chromosomes formed into an X shape. Again, those are the keys we're looking for in prophase. Now, each chromosome has been replicated to form a double structure consisting of 2 sister chromatids joined by a centromere, just like in mitosis. So we have those X-shaped chromosomes. So chromosomes come together in those homologous pairs called a tetrad. And each pair is composed of 4 chromatids. This pairing of homologous chromosomes is called synapsis. And the chromatids can intertwine, and they'll sometimes break and exchange segments of DNA. So during meiosis, we have much more exchange and transfer of DNA between the different chromosomes in a process called crossing over. And this allows genetic material to be exchanged between homologous pairs. And this only happens in prophase and metaphase 1 of meiosis. So those chromosomes will overlap when they're really close together and move genetic material from one to the other. So when we look at those tetrads lined up, we might say we have those 2 tetrads from the sperm, 2 blue tetrads from the sperm, 2 red tetrads that you would have gotten from your mother, from the egg. Now, as they've lined up on the equatorial plate, or they've lined up together, we can see the corner of the blue and the corner of the red are touching, and some of the genetic material was transferred. And now it becomes separated from its other tetrad. So this is known as crossing over, the genetic material going from one chromosome to a different chromosome. This only happens during meiosis. So in metaphase 1 is when crossing over happens, because those homologous chromosomes have attached themselves to spindle fibers and lined up along the equatorial plate as a tetrad. So metaphase 1 looks different from metaphase in mitosis because we have tetrad pairs on the equatorial plate instead of just individual chromosomes. So that's the difference, and we still have those spindle fibers attached to the centromeres. In anaphase 1, homologous chromosomes move towards the opposite poles in a process called segregation. Now, this is where our reduction happens. We went from a diploid organism to a haploid, or a diploid cell to a haploid cell. So we have 2 copies of each individual chromosome, and they're separated into 2 copies of each. We have 2 copies of all of our homologous pairs, and now each cell is going to have 2 copies of one of those homologous pairs. So the chromosomes were made double-stranded, but they are now haploid cells. So we have only 23 chromosomes, even though they're double-stranded, in each cell after anaphase 1. So anaphase 1, the tetrads separate, and we've gone from a diploid cell to a haploid cell. So the parent cell was diploid, 2N, and now the daughter cells at the end of anaphase 1 are haploid. So in telophase 1, that cytoplasm is going to now divide. 2 cells are formed. That nuclear membrane is going to form around the chromosomes, which each of those in each of those 2 cells. But these cells, unlike mitosis, are no longer identical. They don't carry the same genetic information as the parent cell. They carry half the genetic information as the parent cell. Now, complete cytokinesis doesn't occur before meiosis 2 because the cell is going to continue to divide. So in meiosis 2, this involves the splitting of the sister chromatids, but our cells are already haploid, and there's 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2. Now, in some diagrams, and like you have in my slides here, we'll see this happening in 2 cells, but some diagrams will still only show you this occurring in 1 cell. So again, in prophase, the nuclear membrane dissolves and the spindle fibers form. In metaphase 2, the chromosomes are going to arrange on the equatorial plate. And the chromatids are still held together with a centromere. In anaphase 2, the sister chromatids are going to move to opposite poles. And in telophase 2, the nuclear membrane is going to form around the chromosomes. There's going to be a second division of the cytoplasm, and 4 daughter cells are going to be produced. Now, in sperm, all 4 of those daughter cells are viable. In ova development, we have 1 viable egg cell and 3 polar bodies because we have an uneven division of the cytoplasm. So looking at the difference between mitosis and meiosis. So on the left, we have our meiosis. We have crossing over and synapse that occurs with our tetrads. Then the homologs align independently along the equatorial plate in metaphase 1. The homologs are going to separate in anaphase 1, and daughter cells that are haploid will form. Those haploid daughter cells are then going to go through meiosis 2 and divide into 4 ungenetically identical daughter cells. Compared to mitosis, which starts with a haploid cell, the chromosomes are going to align individually along the equatorial plate in metaphase. The centromeres are going to split, and each chromosome is going to be separated to the poles in anaphase, and during cytokinesis and telophase, identical daughter cells to the parent cell are going to form. So there's our breakdown of meiosis, the forming of gametes, and comparing it to mitosis. Take care. Bye.